All right, welcome at this DASH webinar concerning Neuroinsights, Unraveling Disease Pathways with AI. First, a brief introduction of the context of this webinar. It's organized by Femke and the Data Science Center in Health in UMCG. The vision is to be an international leader in data-driven healthcare innovation and everything that comes with that. And why do we want to do that is to basically bring data-driven innovations to our patients here in the UMCG, building the future of health and transforming health. And this is done in multiple ways, actually, both activities as well as facilities. But what's important for you, and hopefully there's many students present here today, is if you want to learn about AI, if you want to get engaged in any scientific projects, please contact us, and we're happy to help you on your way and get in touch with the area of your interest. And we're doing this by sharing knowledge, and then their experts are part of this community, sharing knowledge and helping students, researchers, and then also clinicians on their way in this super interesting field. What we'll do is today, since we have a little bit more time than usual, our distinguished guest speaker, keynote speaker, allowed you guys to interrupt. So please do so by using the chat. I will keep an eye out on the chat, and we'll interrupt Inge whenever there's a question coming up that may needs answering. Our keynote is Inge Holtman. She's an assistant professor here in UMCG. Welcome, and thanks so much for being here. She's very established. She got her Bachelor of Psychology here in Groningen, and did a master research thesis in Montreal after doing her research master also here in Groningen. Then she moved on to become a PhD student, and she did that with honors, cum laude, also here in Groningen. And then she moved to UCSD in San Diego for a postdoc fellowship, and she did that for 3 years. It's 22 to 24 degrees every day there, so I'm very curious to hear why she on earth came back here to Groningen. But she did with the Veni scholarship, super prestigious as well. I did not manage to get that, and I think I was in the same round with you, so thank you for stealing that one. And then she is now a Rosalind Franklin group leader, and got over 5 million euros in grant money. And I love the fact that you still mention the Hendrik Müller Vaderlandsfonds and the Marco Polo travel grant of 625 euros in that list. But also for students, it's very worthwhile to apply and start small. So without further ado, she did get her work into Nature Medicine recently, and that's part of the subject today. Inge, thanks so much for your time, and please take over the controls. Thanks for being here.
Thank you so much. It's a pleasure. Thank you for inviting me to speak. I want to thank the organizers for allowing me to speak here and to talk a bit about my work. Always a pleasure. And you've already mentioned that it's fine to interrupt. You can interrupt by typing something, but also we can make it very flexible and informal. So if you just want to use the mic directly and ask me a question, why not? I don't think there's 500 people here, so it should be manageable. But it really depends on what you want to do. I think on a conceptual level, where I always like to start talking about is the fact that, if you take a random group of people, it's really difficult to determine who will get a brain disorder. There's many different kinds of brain disorders, and we know that many people in their lifetime will develop one. Like it's either Alzheimer's disease, multiple sclerosis, psychiatric disorders. It doesn't really matter. For all of these disorders, the same sort of mechanisms apply. Is that we don't know who's going to develop a disease. And if a disease is being diagnosed, it's extremely difficult to tell how the disorder will develop. So both diagnosis and prognosis are still, well, suboptimal, let's say. And I think there are a couple of reasons for that. First of all, GWAS studies and exome sequencing studies and many other kinds of studies have identified genetic features associated with these disorders. But how exactly do these genetic predispositions influence the neurobiology? That's an active field of research, and there's still a lot that we don't know. Similarly, a lot of ongoing works try to identify what are the cellular and molecular drivers of different disorders. However, different disorders have different neuropathological axes that are partially shared between these disorders and partially disorder unique. And exactly how that goes in a sort of a cross disorder way is also currently not completely understood. And it also entails the third point that I think is always very important to mention here is that the relationship between the clinical manifestation of a disorder and the underlying biology are really not one-to-one. We see that if you open up a person's brain by performing neuropathological examinations, that often quite a different picture emerges than what the clinicians were thinking during their lifetime. So, for example, for many dementias, it's maybe like a 20%, 25% misdiagnosis rate. And that's really high. And it probably is because, where the disease strikes and how the disease manifests are really not always the same thing. And in order to address these kind of questions, the Netherlands Brain Bank has been pivotal on a global level. I'm also affiliated with them, so I have a double affiliation in UMCG as well as Netherlands Brain Bank. And I think it's important to mention what the Netherlands Brain Bank has been doing. It was established in 1985 by Dick Swaap and has an extremely short post-mortem delay, on average only 6.5 hours. And you can imagine that, for brain tissue, that is really vitally important. And the Netherlands being a small country, it's able to get these numbers down. If you compare this to like brain banks in the States, for example, many of them have like 24 hours, 36 hours, or even more. And the Netherlands Brain Bank has been doing more than this number is already outdated. It's now more than 5,000 human brain autopsies since that time. There's a lot of different disorders. So there's control individuals, there's Alzheimer's disease individuals, there's a psychiatric cohort, multiple sclerosis, frontal temporal dementia, progressive supranuclear palsy, as well as many different psychiatric disorders. And the number of registrations is even higher, and the Netherlands Brain Bank just like keeps on going. And what they're doing is they dissect these brain, they take the brain tissues from these individuals after death, and that's all being stored into these freezers that are extensively characterized for brain tissues and are disseminated across the world. So tons of research groups in the whole world are making use of this resource, a resource which has been vital for dozens of yearly publications. But on top of that, they also obtain a lot of information. So they create these sort of like clinical neuropathological summaries. And on one hand, they go back to the to the medics, the GPs, the hospitals, the people treated during their lifetime. And that contained a lot of information. It's all written down in the form of these sort of like text formats. So we know the age, we know the sex, we know postmortem delay, what kind of diagnosis were these people given, what kind is this sort of clinical history file that describes a lot about signs and symptoms, intoxication, what kind of alcohol, smoking behaviors, family history, and a lot of other data types as well. And they also perform extensive neuropathological examinations. So neuropathologists really go through different brain regions and very extensively write down what's happening. It's at the global scale. So we talk about the circle of Willis, for example. How do the blood vessels look? How do the ventricles look? But also on a microscopic level, like how do different brain regions exactly look? Is there gliosis and dystrophic neurons? And this goes up to like 70, 60, 80 features, depending on which specific brain region we're looking at. And all of this information together is being summarized in what we refer to as a main diagnosis, which is a final neuropathological diagnosis. And we basically thought like there's such a wealth of information here, maybe we can really start drilling into this. That was the idea we came up with, me and the director of the Netherlands Brain Bank back in 2019. And the idea to systematically start mining this beautiful resource. And the name of that project is the Netherlands Neurogenetics Database. And in the Netherlands Neurogenetic Database, we basically have four main branches. So we've performed genome, we performed Illumina SNP chips to get an assessment of people's global common variant structure. We've been processing this clinical medical history, like using an extensive set of parsers as well as large language models. And we're also now implementing these large language models in the context of neuropathological data. And with the recently admitted gravitation proposal, we're also going to be performing extensive multi-omics profiling from a large number of individuals. So it's really going all the way from the neuropathology up to the clinical phenotypes and all the underlying neurobiological processes. So that's really kind of a cool endeavor, I think. I'm really, we've been working on this for a number of years now. And obviously, I don't have time to discuss everything we've been doing, but I will mostly focus on this part, the clinical and the medical history. And yeah, this is, as Job mentioned, this was published like 2 months ago in Nature Medicine. Nienke Mekkes is the first author on this project, but a lot of people have collaborated on our side as well as on the side of the Netherlands Brain Bank. And here you can see sort of a workflow of this specific project. So the project had like 4 main phases, let's say. Oh, in this figure, we actually made it 3 phases, but the previous figure had 4 phases. It's always like how to determine a phase, I guess. But the first phase is that we have the clinical and the neuropathological attributes that we were interested in. So we have these the NBB donor files that I was describing before. They contain a lot of information, like the neuropathological diagnosis, so as clinical diagnosis per year, as well as this clinical history that describes the disease progression. And like when we were going through it, at first I was doing this myself, basically. I'm not a clinician, but I was kind of like, what's actually in this data? So I was like, oh, looks like the person was depressed. So showing depressed moods. And we basically went through it, like what kind of like neuropsychiatric relevant signs and symptoms are in there. And this list kept on growing. So at first I identified the number myself and I started labeling a couple of sentences and tried to see if I could like, build some AI models to identify those. And that worked pretty nicely. So we basically extended this, and at some point, multiple collaborators identified more than 90 cross-disorder signs and symptoms, which in the end, 82 seemed valuable. And then we labeled a whole set of them. So we labeled more than 30K sentences. So individual sentences describing whether a neuropsychiatric sign or symptom was observed, yes or no. We also built an ontology structure because then you have all of these signs and symptoms. What do they mean? So we kind of like made major axes such as psychiatric, cognitive domains, motor domains, sensory domains, but a much more fine-grained structure to sort of encompass how those signs and symptoms are related to each other clinically. And that created in the end these sort of data sets, what we call clinical disease trajectories. So we went through a number of phases, and these trajectories are sort of binary matrices. On one hand, you have years, and at the other axis, you have signs and symptoms. And for each donor, you have a matrix like this that describes which signs and symptoms were reported in which years. And well, these are kind of unique sort of data, right? Like these 3-dimensional matrices, and you have many of them for many donors, more than 3,000 donors. But then the question was, once we had this, like, what do we actually do with this information? And we started like playing a lot of like data science tricks, right? So we compared this to the clinical diagnosis, right? Were the clinicians right? Were they wrong? And they have a lot of these sort of ambiguous categories. And we build like AI models to try to see, can we predict the neuropathological diagnosis given a particular set of clinical input information? Or can we, use dimensionality reduction approaches to identify clinical subtypes? So just let the data speak for itself. Yeah, I'll talk about each of these approaches a bit more in depth. But here you can see, the psychiatric symptoms that we finally identified. As I mentioned, we have a very interesting ontology structure behind this. There's much more higher level resolution on how these symptoms are related to each other. But on the global level, we identify these 5 broad domains. And then within those, we have different groupings, such as disturbances in mood and behavior, changes in consciousness, etc., etc. So we presume that these kinds of symptoms are really important to discuss, to study different brain diseases. There was a lot of ontology structures and such focus on what specific disorder, like dementias, or specific one for multiple sclerosis, or there's the DSM for psychiatric disorders, but there really wasn't a structure that we could use to go for like a completely cross-disorder approach. So by itself, identifying these features and defining them, that was already a lot of work. And Minke Groot from the Netherlands Brain Bank has done a lot of work on that, among others. Well, then to go back to the outline I showed before. So we labeled these sentences, we defined the signs and symptoms, and then we set up this natural language processing workflow. And that's where Nienke really came in. So, we go through multiple phases. First, there's a model optimization stage. So we had to clean our training data, which was like 20,000 individual sentences. And a subset of those were used to train and validate, like 15,000. We did like a cross-fold validation structure in which, different subsets were being made, 5-folds. And ultimately we used Optuna to perform hyperparameter tuning. And we compared a number of different models, right? At this point, ChatGPT and related GPT-based models were not yet out. In the end, we have tried to implement those as well, but it didn't really give a lot of added value to what we've already done. But we tried different models, more simple ones like SEMs, a bag of words, and also like BERT, which is basically the model that Google was using for its search engine. And then some of those BERT models were retrained. So this one was refined on PubMed, for example, or this BERT was refined on a lot of like biological and clinical information. We also used T5, is another model from Google. So we were basically using a lot of different models, retrained them, test them systematically to assess which one was best, and then ultimately identify what is the best overall model, retrain that onto the full corpus, and then use that for the total set to identify what signs and symptoms were identified in which specific sentences. And we also selected only the high performing attributes. So we noticed that a couple of signs and symptoms just did not work well. They were too vague, they were not common enough. And in the end, we had a set of 82 different neuropsychiatric signs and symptoms that we could reliably predict. And we also knew when these signs and symptoms were occurring, right? So as I mentioned before, the data was parsed per year. So on a year level, we basically determined which sign and symptom were happening here. And it was kind of funny because, from a data science perspective, we just thought like, it's a kind of cool data set. It's interesting. A lot of clinicians were a little bit like, critical, let's say at first. They were sort of like, yeah, what does a year actually tell you? Right? Did they have the symptom only one day? Did they have the symptom like the whole year? And I fully understand the point, right? Like we don't know. We just know that it was observed into the medical summaries that were generated by clinicians. But, this is the level of resolution we have. And I think it's the same with any kinds of data. If you have microscopy data, you have limited resolution. If you have genomics data, there's always a lower limit of your detection. And for us, the lower limit is years. But interestingly, because we have these medical summaries, they sometimes really went back like 30 or 40 years. So even though maybe the resolution in the time frame is not so much, because with these really long periods of time, it actually turned out to be quite a nice resource. And we can use in all of these different settings. And but the first question people were asking, and we were also asking ourselves once we had this sort of final model, like how reliable actually is the data? And what can we do with this? And one of the ways we looked at this is using this sort of dot plot visualization. And this integrated dot plot is like a lot of like things are happening here simultaneously. So at the y-axis, you see different psychiatric symptoms, a subset of them like restlessness, mania, depressed mood, compulsive behavior. And here you see the number of observations. So the bar plot here corresponds to the number of times this symptom was observed. At the x-axis, you see different diagnoses. So we see control cases, the Alzheimer's disease cases, Parkinson's disease, etc., like vascular dementia, multiple sclerosis. And here are more the psychiatric cases like major depression, bipolar, schizophrenia. And we basically now compiled each of these the symptoms over these different disorders and then performed a permutation test to assess whether a specific neuropsychiatric symptom was more frequently observed in a specific diagnosis than you would expect by random chance, let's say. And then the size, so that if it was significant, you get a star in the middle. The size corresponds to the percentage of cases. So if it's a really big dot, then almost 100% of cases observe this. If the dot is relatively small, maybe like 25%. And there's a circle around it, which was like identified by a clinician. So clinicians would assume which symptoms do you need to diagnose a particular disorder or are frequently used for diagnosis. So that's another way that we can sort of test how many of the circles that contain stars or are significantly overrepresented also contain a circle, which is sort of like used as in the global enrichment score to indeed validate whether our approach was resulting in biologically, medically meaningful information, let's say. And that turned out to be the case. So if you just look at the one that I highlighted here, like mania and depressed mood, those are very obvious, right? But the mania is really not observed by the model pretty much anywhere. There's maybe a few cases of frontal temporal dementia that have it, but not significant. A few cases of major depressive, a few schizophrenic cases, but really low percentages. And pretty much everybody who was diagnosed with bipolar disorder had mania observed into their medical summaries. And also, on average, it was observed many different times. So it was not just 1 year, it was a bunch of years in which this particular neuropsychiatric symptom was observed. But that's really what you would expect, right? Mania is a symptom that is really, well, I guess a core symptom of bipolar disorder. So that it came out so strikingly, it was really a nice proof of principle for us. And I can talk about this a lot, right? The same with depressed mood. Depressed mood is quite common in a lot of different disorders, but obviously it's most common in cases that were identified as major depressive disorder or bipolar disorder cases. But you do see that a considerable number of patients with other disorders also tend to have major depressed mood. And I think that's also one of the importance of this study, because this kind of phenotypical data in postmortem research is really at this point in time not studied at all. Right? People look at like post-mortem tissues and they compare, for example, Alzheimer's disease cases to control or multiple sclerosis cases to controls. They're trying to understand what are the molecular features that differentiate them, what are the pathogenic mechanisms, but pretty much no one currently is like incorporating more extensive phenotype data. And presuming that like all Alzheimer's disease cases are the same or all multiple sclerosis cases are the same is a simplification that's just not correct at this point in time. There's a ton of heterogeneity in how these people are manifesting these diseases. But because brain banks pretty much never have this kind of information available, right, it just wasn't possible to do this kind of work. And I think that's really one of the big novelties of this study is now we have so much clinical information from these donors that you can like start studying all kinds of like neurobiological questions that you couldn't really not address previously at all. So that's a small part of the data set, and here's a bigger part, right? So now we also see the different domains, like the sensory domain and motor domains and all the different subgroupings in those general signs and symptoms such as stress and sleep disturbances, migraine, fatigue. Fatigue is a really interesting example, right? We all know that a considerable number of patients with multiple sclerosis are developing fatigue. No one knows what is the neurobiological mechanism underlying this fatigue. But now that we're able to identify this as a significant enrichment in multiple sclerosis, you can really hopefully move towards identify what differentiates patients with MS who develop fatigue versus patients with MS who do not develop fatigue. Can we pinpoint what the upstream factors are? Somebody's asking a question or somebody has their mic open?
I'm afraid it's a ladder. Please close your mic.
Okay. So we have a ton of information. It's a wealth of information across so many different domains. So it was really kind of cool when we had these first plots and we showed these massive enrichments, really in a disease-specific manner, right? All of these extrapyramidal signs popping up in Parkinson's disease and Parkinson's disease with Lewy bodies and progressive supranuclear palsy cases, and really in sort of a disease-specific way that we can identify what's happening here. But this data, at least in this particular visualization, is really flattened, right? So we just compiled a number of observations and throw out the temporal component. And then we also did a lot of work to see like to what degree can we use this temporal information as well. And that's depicted here. So if you, for example, look at a symptom such as dementia, so dementia is a group of disorders, but it's also a symptom. And if you look at this symptom, how often is it occurred, right? What's the distribution of the observations over the different disorders? You see that like it's mostly happening in cases who are diagnosed with frontal temporal dementia or Alzheimer's disease, which are obviously two major dementias. So there's already, there's a lot of, they're much more significant in these cases there. But we also see that these temporal information are also giving quite a bit of information. Like the frontal temporal dementia cases tend to be much earlier, but there's a lot of spread, right? There's very early cases as well as very late cases. For AD, there almost seems to be like sort of a double edge. So there's a little tail on this side and a tail there. Makes you wonder, like, could there be like a subset of early cases and a subset of late cases? And we also found some evidence that maybe these ones are more APOE4/4 enriched, which is now in line with the other, that's sort of a subset of a subtype of AD. Yeah, and in the same way, we could do like a survival analysis where you try to see, well, if somebody has a particular diagnosis or a particular symptom, how long do they still tend to have? And we see here, for example, that cases with FTD and AD have quite a few years to go after they were first diagnosed or have first show a symptom such as dementia. But for some of these other diagnoses, like Parkinson's disease with dementia, it just goes much faster. And so it also kind of helps to partition things like whether the same pathological features are underlying these different disorders. And we did this for a lot of different symptoms. Like here we see the same sort of, analysis for bradykinesia. So the one at the top is really, very much in line with what you'd expect. There's no biomedical novelty here. So this is very much like proof of principle. But if you use a symptom such as bradykinesia, which is one of those extrapyramidal signs, which is quite common in Parkinson's disease, dementia with Lewy body, multiple systems atrophy, and progressive supranuclear palsy, we could kind of ask the same question, right? Like MSA, PD, and DLB are all driven by alpha-synuclein. They have the same sort of neurobiological disease, let's say. At least they have shared features. And the question in the field has been already for a while, like, is this MSA just an earlier form of this alpha-synuclein, or is it really a qualitatively different disorder? And our analysis is nicely showing that the PD and the PDD case are extremely similar. So whether you have Parkinson's disease with or with dementia, it doesn't really matter. It's pretty much the same disorder. And, maybe the dementia cases are just slightly earlier with when their dementia strikes. But MSA and DLB and PSP are really quite uniquely different in their temporal development, which I think is sort of an indication of what's happening at the neurobiological features. But that's obviously a hypothesis that we need to test into more detail. As I said, we also assessed the diagnosis accuracy. So, for example, here you can see for control cases, like out of all of the control cases that Niels Brainbeck has identified, are they always control? And we see that there's also a little bit of a discrepancy there, right? Like in 90, there's a small subset of individuals that are neuropathologically a control, but the clinician did think that they had a neurodegenerative disease. So that's interesting. What is happening with those 6 individuals? It's not many. But at the same time, you also have a large group of cases, or 91 individuals, where the clinicians would say, well, these don't have they are controlled, but the neuropathologist doesn't really agree. So they do have some, neurodegeneration ongoing. And in the same way, also for like Alzheimer's disease, we see that there's, the majority of cases are shared. But as I said, like, 84% of cases that were neurodegeneratively associated, 16% of those did not have a clinical diagnosis of Alzheimer's disease. So the clinicians, tend to miss this subset. Maybe they think it's frontotemporal dementia. Maybe it manifests as Parkinson's disease. From this analysis, that's not really clear. But at least the clinician did not seem to think that this was an obvious case of Alzheimer's disease or a generic case of dementia. And the
Inge,
same,
may
yeah,
I ask a question? I think it's a good timing for one now.
sure, yeah.
So great presentation, thank you. And you show a heterogeneity in symptoms. I was wondering about the heterogeneity in clinical diagnoses. So we tend to say surgeons agree mostly with themselves, but not so much with each other. So if you have 12 surgeons, you have 26 diagnoses for the same patient. How is that accounted for in this database? I can imagine that maybe one psychiatrist diagnoses a patient, or maybe 20, or how does that work in the brain bank?
Yeah, no, that's a really good point. That's a difficult problem. So in the end, we just took all of the different diagnoses and we basically made sets to identify whether a diagnosis is sort of correct, it's really clearly incorrect, or it's sort of ambiguous. And so for Alzheimer's disease, for example, we see that in the Netherlands, like so often the medics are not really specifically using the term AD. They just say dementia. So we said that if the clinician says it's Alzheimer's disease, or if the clinician said it's dementia, we will presume it is it's good enough, let's say. So the cases where the neurodegenerative disorder and the clinical diagnosis are mismatching is really none of the clinicians were picking up on this really striking Alzheimer's disease dementia signature. So they were either all like saying frontal temporal dementia or Parkinson's disease or whatever. So, but if like, we were lenient, because if you have like indeed 10 different opinions, it's kind of like, yeah, if only one of them is correct, then we would presume that it's still relatively correct, let's say. But it also depended a bit on the temporal profile as well. So if I think an early medic would say Alzheimer's disease, but maybe 10, after that, everybody would say frontal temporal dementia. And the case the person did turn out to have Alzheimer's disease, we would consider that a wrong diagnosis. But it's a really good question. But this is really like fine nitty-gritty use, because it's kind of hard to directly assess. But most of the time when we, some of the clinicians were right in a person, we would generally say that it goes towards the middle category, let's say. So this is quite a more, the serious cases where a lot of most of the clinicians were wrong. Almost all of the clinicians were wrong. And you see, that's a little bit disorder specific, right? Like vascular dementia is like, I don't even know what to think about it. It's completely all over the place. It seems that there's very little congruence between vascular dementia neuropathologically and clinically. But also for frontal temporal dementia, we see this is the case, that quite a considerable fraction are missed. Well, but then for a disease like MS, for example, almost always it goes right. There's a few percent where you'd say there's a misdiagnosis, but that could also be that we have a bit of missing information. Yeah, so we play the game of the diagnosis accuracy. And as I said, we also try to play a game. Can we predict the neuropathological diagnosis based upon the clinical disease trajectories? And for that, we implemented a model which is called GRU-D. It's a gated recurrent neural network that's basically developed to deal also with missing data, and also has a temporal component to it. So we had this temporal component. We weren't sure what to do with it, and also we know that there's missing information. We don't pretend that we know everything about all of our patients. And this model has an architecture that's specifically suited to deal with it. And again, we had like 18. In this situation, we used the smallest set of donors, because for some of them, they're just too ambiguous in their diagnosis, right? If somebody has multiple dementias or vague dementias or rare disorders, we couldn't really use that for this analysis. So we only focused on, relatively common disorders, let's say. In the end, a subset of 1,810 donors. We again did a cross-fold validation. And in the end, yeah, well, optimized with Optuna, and then assessed which one did best with different model architectures. And in the end, we compared this between the clinical diagnosis, the neuropathological diagnosis, as well as the model prediction. Well, this is basically a more extensive version of the figure we were looking at before. But now you can, for example, see that, the GRU-D, overall, the largest number of cases with Alzheimer's disease are detected by the clinicians, by the clinical, by the neuropathologist, by the clinical diagnosis, as well as by the model. But there are differences as well. Sometimes the model and the clinicians agree, but it's neuropathologically incorrect. Sometimes the model is better able to identify the neuropathological disorder, but sometimes the clinicians are better. So it seems that it's a bit on par, right? It wasn't like, especially for the more common disorders like AD and MS, and like I think also for frontal temporal dementia, it was relatively on par. Maybe the clinicians are already a bit better at frontal temporal dementia, and there seems to be a correlation between the number of observations, right? So if you have a disease that's common, like Alzheimer's disease, we have a lot of them, then it's relatively good at identifying them. The same with Parkinson's disease or MS. But if you have a disease that's quite uncommon, like multiple neuron disease or so, yeah, the model pretty much never assesses it. In a few cases, it does. So I think this is a simple situation where, for reliable AI models, you just need a lot of observations. And I don't think we have sufficient observations, and also the data is maybe not, it also the model is struggling, right? Because this massive amount of heterogeneity between these disorders. It's not like the model was able to do this perfectly either. And yeah, just we wanted to identify more about like what does the landscape of the heterogeneity actually look like, right? So and then we use the same data set again to identify clinical subtypes. So we established a dimensionality reduction framework that's able to deal also with the temporal component. And well, this is sort of what the UMAP and the K nearest neighbor clustering in the background. That's like identified 6 main clusters. And this little structure came out, and some people tend to see a brain into it. I personally don't, but some people thought that was funny. But at least what you can see here is there's 6 main clusters, and we identified labels to those clusters. And on the right side, you can see how the clinical diagnoses are sort of like overlapping with these different clusters. So here, for example, you see that a large number of Alzheimer's disease cases tend to fall into this late dementia, while a lot of frontotemporal dementia cases tend to fall into this early dementia. But you already see the complexity here, right? That there's definitely FTD cases that are in the middle of the Alzheimer's disease cloud, and there's also a lot of Alzheimer's disease cases that are clearly in the FTD cloud. So it makes sense that clinicians are not doing this perfectly and that the model isn't doing this perfectly either. And it's just in line of this tremendous amount of heterogeneity of these disorders. It's just really difficult to, from a clinical perspective, fully understand what the neurobiology looks like. Another one I find interesting is here, like the psychiatric cases. Those are in yellow, and then here we see the control cases. And we see that the psychiatric cases are here, the green ones, and the control cases are the yellow ones. And this is sort of in line, I think, with this like gradient approach, right? Like, you cannot really say most of the individuals were sort of on a line of a continuum, right? Like most cases with psychiatric disorders aren't really black and white. And that's also what comes out here. Like in some situations, these yellow people up there, I presume they look more like psychiatric cases than control cases based upon, the kinds of symptoms they manifested through life. And in the same way, also with this cloud of multiple sclerosis individuals, we also see that there's a little bit of an overlap. Those are probably the very mild cases of MS. Yeah, and what is also kind of interesting, the next thing that we want to do here, and you can already see that from the shape, is that we want to see like you have the correct diagnosed individuals, but also the incorrect diagnosed individuals. And is that random? Is the incorrect diagnosis, are those just all over the place? Is it just like, you have an Alzheimer's disease label, but the clinician, or you have the neuropathology of Alzheimer's disease, let's say, but you are sort of atypical. So can you wind up everywhere? Can you look like an MS case and like a PSP case and a late dementia case? Or are you sort of like systematic? Are there systematic sources of variation that underlie this misdiagnosis pattern? And that's also what's depicted here. So we have these yellow colors, the white colors, that mean stars for general overrepresentation. So the late dementia cases, for example, are overrepresented for Alzheimer's disease. But then the black stars are meaning whether a diagnosis is, whether the clinicians were wrong with these cases. So those were inaccurately diagnosed. So are the misdiagnosed Alzheimer's disease cases random? Well, it turns out that's not really the case, right? So a lot of like Alzheimer's disease cases that are misdiagnosed actually wind up in either the Parkinson's cloud, let's say, or sort of in the asymptomatic control cloud, which is kind of interesting. That's not what we expected. Like I was expecting that they would sort of pop up in the early or in the late dem, but that's not the case. Alzheimer's disease is enriched in both of those, right? That shows this small subset of Alzheimer's disease cases, they just manifest their disease completely different than what you would expect. And in the same way, in the control asymptomatic cases, there's a small set of individuals that's actually popping up in the late dementia. Again, interesting. So yeah, we found a number of these sort of like weird cases that we wouldn't expect, like PSP. PSP tends to be part of this sort of Parkinson's disease cloud, is the one here at the bottom. And then we see that the cases that are misdiagnosed, that progressive supranuclear palsy, actually tend to wind up in the dementia cases. So they manifest as, quite obvious dementia cases. So the clinician would just say, well, you have dementia, Alzheimer's disease, or frontotemporal dementia as such. But, from a pathological perspective, you actually have something quite different. Yeah, we made all of this data available on our website, nnd.app.rock.nl. We're moving now towards like a Google Cloud-based environment. So I think we're also going to try to get a new URL. But for today, so yeah, again, there's much more I can talk. And maybe, if I'm invited to speak some more, I can talk a bit about the genetics as well. We are now doing like a genetic analysis. We're also working at the neuropathology. But to conclude for this part of today, I think that data-driven approaches are very powerful methods to disentangle the heterogeneity of brain disorders. This is really necessary if we want to improve the diagnosis as well as the prognosis. I think we've identified clinical endophenotypes. I only looked at the major clusters, but within each of these clusters, we also performed subclustering, and there we found quite interesting subclusters as well. We looked at the atypical misdiagnosed donors, and that's not random, right? If cases are often misdiagnosed, they tend to share commonalities with each other. So diseases can sort of masquerade as another disorder, which I think is a kind of an interesting finding. And yeah, this is an ongoing work. So we're working with genetics data. We have genetic data for almost 3,000 individuals. We're calculating polygenic risk scores. We're using this data also to refine GWAS studies, because GWAS studies are often also suffering from the same contamination, right? In Alzheimer's disease, GWAS probably has 20% misdiagnosis cases. So that would seriously impact some of the loci identified. So we're trying to see, can we use our genetic data to sort of refine the current GWASs out there? We're using multiomics data and also for prediction in the context of prediction. And as I said, we're using also large language models and neuropathological text data. So that's a lot of ongoing work. Yeah, a number of people in my group have been working on this, like Nienke Mekke, Sander Bouwman, a new PhD student, Ekaterina Dachmanskaya, under supervision of Dr. Bart Egge. I also want to acknowledge my collaborators at the Netherlands Brain Bank. Minke Groot has done a lot of work. And it's always great to work with Inge Huizinga, who's a big inspiration for me and a close collaborator. And I have a number of funding sources I'd like to acknowledge here as well. And yeah, I want to thank you all for your attention.
Thanks so much, Inge. Very impressive. I think you also had a final slide of students who can get back to you for projects, right? That's right. Yeah, that one.
Yes, thank you. I forgot about that one.
Awesome. So we'll open the floor to discussion and questions. I will keep an eye on the chat. For now, please raise your hand if you have a question, so I can pick that up and give you the word. Bert, please go ahead.
Inge, nice work. I have a question, but I might have missed the information because I logged in a bit later. You say this information is important, this genetic profiling is important for diagnosis and probably also for therapy, to tailor therapy on the right disease phenotype. But for that, you would need a genetic profile in a tissue that you can image during the life of the patient. So to which extent do you expect that genetic profiling can be extended to more readily accessible compartments during the life of the patient?
Well, thank you for the question. Yeah, and I mean, I get your point. I mean, the genetics data we're talking about here is really like the fundamental DNA, right? It's just common variant. So we're not talking about transcriptomics or multiomics here. So I think for like that kind of data, it really doesn't really matter that much where it comes from. Like you can get reliable genetics from a nose swab or whatever. Whether like that kind of information is going to reach the point where it's clinically meaningful, I don't know, right? It's an ongoing work. Like polygenic risk scores have potential, but will they be good enough? I don't know. I think no one at this point in time knows, and it's an active field to study, let's say. But yeah, I wouldn't be surprised if that is going to happen in the next number of years. If it's going to turn out that these polygenic risk scores or maybe some individual variants could be worthwhile for diagnosis or maybe even for treatment. Yeah, then I presume that it's very easy to generate those.
But have you done a pilot to see if these profiles are mirrored in, for instance, in the blood?
Yeah, again, like I'm writing a proposal right now to focus specifically on blood profiles. So, we're going to generate a lot of like brain specific multi-omic signatures. That's an ongoing thing now with the gravitation proposal, for example. And I have written a number of proposals where I try to also get blood tissues from those same individuals, because I do think it's very worthwhile to see, can you predict, multi-omic signatures in brain from blood? Let's say, can you predict the neuropathology from blood? So that's definitely something I would like to do. It's on my radar, but at this point in time, that's not something I've done yet.
Okay, thanks.
Thank you. Any more questions? Please raise your hand. In the meantime, I have one. I'm seriously um, challenged here in this topic because I'm both colorblind as well as an orthopedic surgeon. But I really got the point, and I really enjoyed your talk. Two questions. So, first is how would you translate these questions to a practicing or your data? I'm sorry, to a practicing psychiatrist or neurologist. What can they get out of this, the clinicians? And the second one is those atypical ones with a lot of commonalities you found. Is this something that is a build-up to discovering new diseases? Because we don't know what we don't know, and potentially you're finding patterns that are new.
Yeah, no, I think both of them are great questions. Like the new types, right? That I didn't show the dimensionality reduction from the paper in that much in detail. I only showed the global one with the global patterns. We also performed for each cloud a subclustering analysis as well, and then we find subtypes, and whether those subtypes are really also responding, generated by completely different diseases. At this point in time, we don't know. We don't have neurobiological evidence yet, but like on another project that I worked on, which is just accepted in Acta Neuropathologica, we did the same kind of thing specifically for the MS cohort. So we tried to sort of like systematically characterize the landscape of the neuropathology of MS, right? Because MS is also this very heterogeneous disorder. There's a lot of different neuropathological features happening, and how do those relate to each other? And we found three main dimensions. At first, I was also thinking like maybe we can actually find clusters, right? So in the earlier analysis part of that specific project, we thought like, oh, maybe we can find subtypes of MS. But I think the data now is showing that you cannot find subtypes of MS, but you can find axes of neuropathology, and those axes together sort of determine where a person is and how severe their disease is like occurring, let's say. So in the same way with the other neuropathological disorders, I think there's a lot of wealth of data that could help to figure better out the landscape of the heterogeneity. Maybe there are hidden subtypes. Maybe there are also hidden axes of neuropathological features. I don't know, and I think that's basically an active field of what we're trying to study. So that's regarding your new type question. Yeah, and the clinicians and the neurologists and the psychologists, what are the psychiatrists doing with this data? Yeah, right. At this point in time, it's still very fundamental. So for a neurologist or a psychiatrist, I don't have anything to share that I think is like changing their work at this point in time. But I do think that, where we're moving with this is that maybe we will redefine the landscape of what these disorders are actually looking like. And yeah, I can imagine that will have, if you identify new subtypes, if you identify axes of neuropathology, maybe that will have a lot of like consequences on how they will be looking at their own patients in future. Because I do think that data-driven approaches are important for these kinds of diseases, and I don't think we are yet at the point that we are doing the job perfectly. So I hope that our work can contribute to that.
Awesome. And in treatment of asthma in kids, the clustering method basically discovered that they react completely opposite to the medication. They all got the same medication, so
Yeah,
it's gonna be super important clinically in the end.
yeah.
All right, I think we are going to round up. I see Michiel raising his
Can I have
hand.
one question?
Yeah, you go
Yes.
ahead, Michiel, and then Bert will finalize our rounds of questions.
Yeah, that was my question in relation to the questions of Job in relation what's in for it for the clinicians. But my question was, there's a lot of misdiagnosis, and can it be of help to reduce the misdiagnosis?
Yeah, we did point out in the paper a number of like one of the reviewers was asking about that as well. So we made some supplemental tables pointing out that which signs and symptoms could be helpful for improving specific diagnosis, right? But yeah, I don't know if how to clinically implement that. I don't know if it's actually valuable. It was a little bit like throw them a bone kind of thing.
Mhm.
I don't know, right? Like, we try to learn more, but I'm not a clinician. I don't know exactly what it's like to have a patient in front of you and to diagnose somebody. I understand it's very difficult, right? I'm in no way trying to say that the clinicians are doing a bad job. I understand that they're dealing with limited sense of information. They cannot look into somebody's actual brain. I
Yeah.
just hope that we can maybe better stratify patients and
Mhm.
maybe, you know, better find out what the neurobiology looks like so that clinicians in future can also do their job better. That's the overall aim, but I don't have a clear solution for clinicians right now.
Yeah, maybe they can structure their questions to the patients better or more, or maybe ask more questions to better sort out what type of, phenotype of patients you have. Maybe that could be helpful for that.
It could be, but again, like I think the main point of our paper is that like sometimes these pathologies just influence the phenotype in such an atypical manner that it just doesn't show. If you have an Alzheimer's disease pathology, but it
Mhm.
manifests as a Parkinson's disease phenotype, like how can you clinically then know that the pathology, the underlying pathology, is actually something else?
Yeah.
Right. So I'm not sure if we can solve the problem only looking at it from phenotypical perspective, let's say. Well, maybe blood can help, maybe genetics can help, and maybe other biomarkers can help.
Anyway, I really like the work. It's very nice. Thanks.
Thank you.
Quick last question and a quick answer, please.
Yeah, it's probably a longer question, but in the invitation it says that you are going to show us the way to the future of neurology, neurological research. And basically I see two main questions, and one is what causes the pathology, and the second question is how does the pathology translate into the symptoms.
Yeah.
These are different questions, huh?
Yeah.
So where are you going? What, in your view, where will be the emphasis of artificial intelligence? Your artificial intelligence techniques. Where will your emphasis be?
Well, I mean, I'm mostly in the middle. Like, what are the axes of the neuropathology? Right. I think that's the first question. What are the axes? Like, the axes of neuropathology are now discovered by neuropathologists who look at a relatively restricted set of features. Right. I'm not sure if the data will fully corroborate that. So maybe we can find better axes of neuropathology that are covering a more wide range of diseases, that are more tailored to the individual person suffering from a disorder. And then maybe that helps to also relate that better to the clinical phenotype as well as the underlying biology. I'm trying to bridge those two fields. So I think it's in the end one question.
Well, the first I understand, but the second I have major doubts. But maybe we can discuss it at some time.
That would be great.
Okay.
Thank you so much for the interactive
Thanks.
and lively discussion. I'm sure that we're gonna have a follow-up session, and to Inge's preferred method, we'll do that live with a live audience, and maybe some snacks as well. Let me round up. Thanks so much, Inge, again. I will give you this virtual bottle of wine, but that will be coming your way, I think, Femke, right? And I'm gonna open my slides now. Just to address the two upcoming DASH events, another online session 18th of June, Transforming Healthcare Education. And please join us live the 27th of June in the Rode Zaal in the UMCG, talking about large language models, but then more in clinical practice rather than research. And finally, please join us. Complete our survey, and then you may win a AI Revolution in Medicine book. So please join us and scan this QR code. I think we're right on time rounding up. Inge, thanks so much. Participant, thanks so much, and we'll see you next round. Thanks.
Thank you.